Okay. Good afternoon, everyone. Good morning from some of you, and welcome to today's webinar in Field Molecular Diagnostic as an Aid to Disease Management, organized by Emphasis Project. This is the second of a series of webinars organized by the project. I'm Silvia Anastasia, and I'm working at Moverim, one of Emphasis partners, and I will be your host for the event today, together with Isabel and Andrea. Before we get started, I would like to go over a few items so you know how to participate in today's event. We have taken a screenshot as an example of the attendee interface. You should see something that looks like this on your home computer desktop. You are listening and using your computer speaker system and your headphones by default. Pay attention to have your speaker's symbol in green, and if you have any problems, please write in the chat or start a private chat conversation with me or one of my colleagues, and we will be happy to help. You will have the opportunity to submit text questions today to today's presenter by typing your questions into the chat of the control panel. You may send your questions during the Q&A session at the end of today's presentations. All participants' microphones are off at the moment, and I can activate your microphone if you need it during the Q&A session. If you raise your hand clicking on the small hand picture at the top row of your interface, I will give to you the word. You can follow our events and Emphasis activities also on our Twitter account, Emphasis Project, and our Facebook page. Later, you can also watch the recording of this event on our YouTube channel. Now, I would like to introduce our speakers of today, Dr. Jane Thomas and Dr. Rosa Caiazzo from NIAB. Some words about them. Dr. Jane Thomas is head of the pathology group at NIAB. She has a special interest in the diagnosis and measurement of plant disease and in evaluation and characterization of host cultivar resistance and how it can be used in integrated control. She is currently working on two emerging disease problems in UK, Alternaria complex in potatoes and Verticillium longisporum in oilseed rapa. She also has a long-standing interest in the seed-borne pathogens of Arab crops, including their detection and importance as a source of inoculum. Dr. Caiazzo, after receiving her Master of Science degree in 2003 at the University of Naples, Federico II, started working on characterization of phytopathogenic fungi at the Agricultural Research Council in Italy. In 2009, she completed her PhD in a degree in agricultural biology and biotechnology at the University of Basilicata. She joined Washington State University for 30 months as an associate researcher in plant pathology, working on fungicide resistance. In 2013, she moved back to Italy and joined the Agricultural Research Council. She has been working at NIAB since August 2015 as a molecular diagnostician. Here at NIAB, she works at the Emphasis Project, developing assays to detect plant pathogens, in particular fungi, and follow the molecular aspect of other projects that involve next-generation sequencing approaches. Now, I give the microphone to Dr. Jane Thomas for the presentation.
So just a brief introduction about the Emphasis project. It's a Horizon 2020 program, and the acronym stands for Effective Management of Pests and Harmful Alien Species and Integrated Solutions. And we're looking at the development of molecular diagnostics which will feed into some of the integrated solutions that are being developed elsewhere in the project. So just the work packages within the project, there are 5 altogether, and we're involved in work package 2, which is the practical solutions for surveillance and monitoring, and that's where our development of in-field diagnostic comes in. So why is diagnosis important? Well, very simply, plant disease management relies on anticipating the occurrence of a disease and a correct diagnosis of that disease to identify the pathogen. And very fast and accurate diagnosis is important to prevent and reduce the amount of damage or economic loss. So why do you need field diagnosis? Why can't you send a sample of a diseased plant off to a lab and get an answer back from them? Well, a lot of the time early infections are symptomless in the field. It's very difficult for agronomists to identify them, and if they send them away to a laboratory, they have often quite a long wait to hear what the problem is. And some diseases have very long latent periods as well, so it's difficult for field walkers and agronomists to even know that a disease is present. And lastly, their disease complexes are very difficult to diagnose, and diseases are not generally obliging. They don't exist in nice simple state. They very often exist with a wide range of other organisms which may also be pathogenic, and accurate separation of that complex is necessary to improve your product choice. And you may be able to select better curative products if you have an established disease, or change your fungicide program depending on what the identification tells you. And finally, potential to switch products if your diagnostic is in the future able to tell you if you have a fungicide resistant population present. Okay, so just a couple of brief examples to outline some of the issues. This is a picture of Phoma stem canker, which is a very important disease on oilseed rape in the UK, and it's caused by two different species of Leptosphaeria, Leptosphaeria maculans and Leptosphaeria biglobosa. And the disease starts off in the autumn as a leaf spot, and that grows systemically within the petiole down to the stem base, and then the following year it produces this pretty serious stem base canker which causes lodging and yield reduction. And in the UK alone it causes around 60 million euros loss of potential yield. The problem is that it's quite difficult to identify which species you have. The leaf spots that both of them produce look very, very similar in the field, and in the future product choice can be manipulated to perhaps give you better control of maculans if that's the predominant disease, or better control of biglobosa if that's the prominent disease. And there is ongoing work in projects outside emphasis that is really looking at this problem and trying to identify optimum products. So when we have that information, it's going to be very important to identify which species you have, which is the predominant species on your leaves in the field, and to do that quickly so that you can get a fungicide program on before the disease becomes established and starts growing down the petiole and into the stem base. So that's one of the diagnostics that we're working on. And a second example with the same crop, this is light leaf spot on oilseed rape caused by Pyronopisiza brassicae. It's favoured by cool and wet conditions, and that causes in the UK about 30 million euros of yield loss per year. It's a disease that is quite important in the north of the UK, but it is becoming prevalent further south and into continental Europe as well. Triazole fungicides have provided good control of the disease, but there is evidence of reduced effectiveness of some of those products. But really the critical thing is the right timing of fungicide sprays and knowing that the disease is present. And it's a very difficult disease to diagnose. It has a very long latent period, and a leaf can look quite green and healthy, and yet there can still be disease just establishing inside it. So another potential use of in-field diagnostics is their application to detecting airborne inoculum, so moving away from the plant and looking at spores in the atmosphere before they infect the plant, and using spore trapping together with diagnostics to give you a forecast of the availability or the presence of airborne inoculum. And PCR assays or lamp-based assays are being used in conjunction with spore traps as a means of detecting available inoculum. And that's very important really, because in the past spore trap strips had to be sent off to a lab, looked at microscopically, very difficult to identify spores of pathogens which look very similar, and that could take a long time, so that speedy application of products was not really an option. So in-field diagnostics to use in conjunction with spore traps is an important development. And the last thing I wanted to talk about just now was soil-borne pathogens. Of course, some diseases are caused by very persistent soil-borne organisms. They can be difficult to diagnose, and detection of pathogens in soil can often be a very time-consuming and difficult process with selective media, qPCR, which does not work very well in a soil background. So lamp diagnostics or isothermal diagnostics can be very valuable in rapidly identifying the presence of specific soil-borne pathogens, and we'll be talking about one later on. So I think that's all I wanted to say at the moment, so I'm going to hand over now to Rosa, who's going to talk about some of the technology that we've been using and some cases where we're applying it.
Okay, so I'm just going to focus on how it's possible to do diagnosis in plant pathology. The first things to keep in mind is the proper identification of the plant, because we know that sometimes different cultivar can be susceptible or more or less susceptible to a specific race. Like, for example, we have wheat that could be susceptible to a specific race of Puccinia graminis. So if we know which one is the plant, we can try to have the right pest management control and try to plant the cultivars that are supposed to be less susceptible to that pathogen predicted to be present in that season, in the growing season in that specific area. Identify characteristic symptoms and signs that are different according to which pathogen is causing a disease. To understand the biology of the microorganism, identification of the pathogen, and all these in order to have the right disease management control on the disease. So in these tables I reported some of the symptoms that can appear on plants. It could be symptoms on leaves, on stems or root, and each one can lead to a different pathogen. And the most important thing is to find the sign of especially fungus or bacteria on a plant. Physiological symptoms, look at the lesion, see if there is any sign of a fungus like mycelium, spore, like mold of rust, or sclerotia. In the two pictures we can see on peronospora, we can see blue mold, and we can see the sclerotia in the stems of Oilseed rape. So look at the symptoms to find a trace of the pathogen that may help you to have a better identification on which could be the disease affecting the plant. Traditional plant pathology was quite slow. It's a kind of slow process because first is meant to isolate the pathogen, make it grow in artificial medium. I'm talking about fungi most of the time. Morphological identification could be made by looking at the structure of the mycelium or microscopic observation of the conidia. But the morphological identification that is done in the laboratory is not always specific because it only stays to a genus level. It could take time, and it's sometimes difficult to differentiate between species that are really close and really similar. Like, for example, if we wanted to identify early blight by looking at the spores, early blight is a disease on potato that caused by different Alternaria species, and mostly is Alternaria solani and sometimes Alternaria alternata, but it's really difficult to distinguish between the two species because first we need to make the fungus to sporulate, and then the spores are quite not always easy to distinguish for shape and different measurement that need to be done at the microscope. But it is important to identify the right pathogen in order to use the right chemicals to control the disease. So we know that the classical plant pathology to identify a disease is quite long because we need to first grow the pathogen, inoculate the plants, and it could take up to a minimum of 6 weeks. And if the pathogen is a slow growing or it's not, and it doesn't grow in artificial media, it could be even more complex and long timing process. So instead, the molecular technologies that have been used lately for diagnosis, they can allow the identification of morphologically similar species and strain, the detection of infection before the symptoms start to appear on the plant, and we can be able to detect the organism, the pathogen, without culturing on artificial media. And this is going to increase the efficacy, the accuracy, and the speed of the diagnostic. And as Jane pointed out before, the faster we act, the better it is in order to try to control the disease. The most used molecular technology for diagnosis are immunological assays that like ELISA, that are particularly used for identification of plant viruses, and nucleic acid-based technique such as PCR, nucleic hybridization, quantitative real-time PCR, and isothermal loop-mediated amplification, LAMP. We're just gonna focus on the LAMP because we try to develop new assay to detect new pathogen by means of the LAMP. And that just before I'm going to introduce for the classical diagnosis in plant pathology, we used to amplify the ribosomal rDNA because it's an essential gene that is present in all the organisms. Is a common target for sequencing study, and especially focusing on the ITS, the interspace transcript spacer, using primers specifically, generic for different fungi. We can be able to amplify a fragment of a gene and then sequence that fragment and analyze in gene bank to have identification of the pathogen. This process is a kind of, it's much more fast than classical diagnosis, but still it takes a week because we need to sequence and make the sequence and analyze the result. So if we focus on different genes and using specific primers that amplify a specific gene for each pathogen that we want to identify using a PCR, but most of the time real time on a LAMP, that is going to make the process much more fast. We have been using for molecular diagnostic basically PCR and real-time PCR, but both of the two techniques, they need specific equipment like a thermocycle, so it's not really handy to do in-field diagnosis or using these two techniques. And plus, the real-time is faster than PCR and more sensitive, but still it needed 2 hours to have the result. Instead, if we use isothermal amplification, we can have a result in 30 minutes, maximum 60 minutes. We don't need to do DNA extraction. We can use tissue samples like stem, leaves, or root. It's high specific and sensitive. It's faster and cheaper than real-time or a normal PCR. So we focus on our part of the project, in the emphasis project, developing a LAMP assay for identification of a pathogen on different crops. Those are the main steps that we follow in order to develop a new assay. That's identification of the specific gene and primer design, optimization of the LAMP reaction condition, validation of the specificity and sensitivity of the assay, because we test the assay on targets and non-target related species or other species that can be present on the same host. We have the design and internal control, and at the end we test on field samples. For primer design, comparing the LAMP to the real-time PCR, we need specific software because we need to design 6 pair of primers. We have free software available online, or if we can buy software available from Permit Biosoft, or in some cases we can just go through the normal laboratory practice, align multi-sequence, and design the primers. This is kind of useful when we try to design assays specific for species that are really close between them, and with the multi-aligner we can detect which one are the mismatch and design more specific primer. As I just said, the LAMP needs 6 pair of primers. I'm not going to talk on how the LAMP assay works because in the previous webinar they already explained very well how the LAMP works. I'm just going to report some of the examples that we have been working on in field and in the lab. This is an example of the Alternaria assay designed to amplify specifically Alternaria alternata. This is the fluorescence result obtained looking at the real-time PCR. We can see that the time of reaction is really fast. We have amplification in the first 30 minutes, and the amplicons is really specific to product because looking at the melting curve, we only have one peak. We focus on some of the most important crop in UK, and as Jane said, we're working on oilseed rape. We focus on trying to develop a say on oilseed rape caused by working on Leptosphaeria maculans and Bicobosa, Pyranopetida brassica, Verticillium longisporum, and we develop an assay to detect early blight on potato that is able to distinguish Alternaria alternata from Alternaria solani. And we also working on wheat, developing an assay to detect transport. So how does the in-field working test? As Jane said before, especially for light leaf spot, it's really important the timing, the early detection of the pathogen, because there is a really short window in which we can apply fungicides in order to control the disease and reduce the loss. So if we go in a field, we just need to cut a little piece of leaves, in this case, because we know that the symptoms appear on the leaves. Do an alkaline lysis for 5-10 minutes, and then using not a thermocycle, not a real-time, but a genie, we can bring the genie in the field, put the tube inside and run the reaction. And then the same way, like in real time, we have a graph in which the fluorescence show us the presence of not of the pathogen. And also, it can give you also a melting curve to see if your assay was specific or not. Another assay that we have been working on is to detect verticillium stem striping caused by Verticillium longisporum. That is a new coming disease in UK. It could cause from 10% to 50% of yield loss. It is present on brassica, but also on other crops like aubergine, lettuce, tomato, and watermelon. We developed a lamp assay to detect the Verticillium longisporum, and we compared this assay with the real-time assay. We analyzed the presence of the pathogen in different samples that they are all supposed to be infected, and we can see that we have the same and sometimes higher sensitive result from the lamp. And of course, compared with the real time, that we have the detection around 30 cycles, that means around 30 more minutes. We can have detection in around 20-25 minutes for using a lamp assay. So it's kind of a really fast result. And as I already said before, we can just run a lamp assay in the field. We don't need a thermocycle, and we don't need high qualified people to do it. In order to test the sensitivity of the lamp, we used DNA extracted from Verticillium longisporum, and here show how 10 times folded dilution give amplification till a minimum of concentration of DNA like 5 picogram. And also in this case, we can see how specific and is the amplification by the melting curve. This table reports all the pathogens that we have been working on in the emphasis project, the gene that have been targeted. The assay have been designed for all the pathogens. We are testing on DNA of the target, non-target, infected samples, like extracting DNA from infected tissue, and also validating the assay, amplifying without DNA extraction on leaves. And we're trying to understand how sensitive could be the assay, how much time before the symptoms start to show on the plant we are able to detect the pathogen for each of the disease mentioned before. So in conclusion, we can say that molecular technology increases the understanding of the biology and the population structure of plant pathogen. It can support disease management decisions that rely on anticipating occurrence of the disease. Because in order to control the plant disease, sometimes it's not possible because there is no any fungicides that can control, but we try to minimize the loss. It's important to avoid the present disease, selecting a time of the year or a site that is not inoculum, so select the right place where which kind of plants or cultivar, specific cultivar we need to plant in order to reduce the loss. Exclusion, try to prevent the introduction of inoculum, eliminate, destroy, or inactivate the inoculum, prevent infection by means of toxicant or some other barrier to infection, use resistant cultivar or at least less susceptible to infection, and cure the plants that are already infected. But sometimes, as I say, it's not possible to totally destroy, so we need to use an integrated pest management control that in order to keep the pest population below the threshold that could lead to economic damage, to economic loss. avoid the problem of pests developing resistance. So trying to use switch fungicide treatment every year, don't use always the same. Using resistant crop varieties
thank
in
you,
conjunction
Jane
with
and
chemical
Rosa.
treatment.
We are now
Crop
going to
rotation
begin
and
answering the
manipulation